that you have to remember a medicine. In mycology, they will not go away unless you do medicine. So it's a different thing. Viruses, they heal on their own. You heal on your own. You have your humoral immunity, you have your cell-mediated immunity to bank on, and you will get cured from the diseases that they cause. Unfortunately, for mycology, not only do you have to use medicine, you have to use the medicine for a long period of time. And when I say long, it is really long. You have to make sure that you have a patient who will be compliant to you. Because treatment for 1 month, wow, are we going to start? By the time we can use the projector, it's already time to go. It's Anthony.
Remind him 3 days.
Maybe just click continue.
Just to continue.
So our topic for today is mycology 1, which includes introduction and superficial mycosis. So there are only 2 mycology lectures. The other one is all about the systematic and opportunistic infections. So the worst ones that you can encounter. Our objective, of course, would like you to get to know about everything about this fungi and get to know more about the superficial mycosis, the superficial fungal infections. And it includes the superficial and cutaneous infections. These are the references for this particular lecture. Let us go first to introduction. Introduction aims for you to get to know the fungi. This is an example of a fungi. Oh, it's rose spelling, mushroom. It's mushroom, no? Without a C. If you take a picture of this and then post it, and then you will tell this is the caravan stories PowerPoint. Well, for your information, I was informed by Dr. Maniwa about your lecture on Tuesday. Alright, there you go. Maybe the original lecturer doesn't want to lecture anymore. Let us wait again for it to come up. So, mushroom. It came from a Greek word, which means mix. No? And mushroom, because the first fungi that they have known is definitely a mushroom. The mushroom that you know, no? That's a fungi. It inhabits every niche of your environment. Even if you think it is inhabitable, for a fungus, it is. No? And there are 80,000 species. 80,000. 400 of these are medically important. However, only 50 species are responsible for your fungal infection. These fungi are mostly beneficial to us. They are beneficial to us. And I know that you have a copy of Dr. Santiago's lecture. So maybe she cannot come because she have to travel just to give this lecture. And that's why it was given to me on Tuesday. So, what are the characteristics of this fungi? What is true to all fungi? One is their cell walls. We got to know about the cell wall that is made up of peptidoglycan, no? Lipopolysaccharide. That's what we know. But for this, their cell walls doesn't have it. They don't have the peptidoglycan, they don't have the lipopolysaccharide. Therefore, what we know about cell wall is not true to this fungi. They don't have glycerol, they don't have the teichoic acid that we usually found, that we found in bacteria, that we found in other of the cell walls. Instead, what they have is chitin, glucan, and mannan. These three structures are the target of our antifungal medicine. No? The antifungal medicine is directed in destroying the cell wall. So that is something that you should know. Another thing is they have ergosterols. They have cholesterol. It is a cholesterol-rich organism. In fact, they love lipids. No? They love lipids. They love lipids so much. So these organisms, just like other organisms, have a nucleus, have the usual organelles. It's just that their cell wall is different because it contains different structures. Chitin, mannan, and glucan. These three are the target of our antifungal medicine. Chitin makes the cell wall rigid. So it's difficult to infiltrate. That is why your antifungal have to break down this chitin to be effective. Otherwise, this fungi will just grow and grow and grow. And as I have mentioned to you, it will take several months of treatment. Mostly 6 months treatment. You have to make that emphasis on the treatment. Because most of us got used to 1 week treatment, only a few days treatment, and then we have to stop our medication. However, this particular organism, the treatment for them, it has to be stretched and maintained for 6 months. Otherwise, they will just lie low and then erupt again. Alright? Which is one characteristic of mycosis. You thought you don't have it anymore, only to find out that it was just resting period. And then you'll have it again. Alright. What else do we have to know about this? This is how they look like under the microscope. This is how they look like grossly. But not all of them looks like this. These are the only ones that we can grossly see. Alright? So, what are the characteristics? They are chemoheterotrophs. Chemo means chemical. Heterotrophs means they have different composition. Alright? So not one fungi is the same as the other. So that's what basically that means. They possesses a nuclei, of course, with a nuclear membrane, and they divide asexually, sexually, or both. So they can reproduce in different ways. That's what it meant. They are aerobes or facultative anaerobes. I am hoping that you know what that means. I'm not here to lecture you about this. Now they grow either reproductively by budding or non-reproductively by hypaltic elongation. So if it is reproductive, meaning they have to grow in number, it is through budding. If it is non-reproductive, just to grow, then it is just deep elongation. Now, most of them are multicellular. However, there are some or few that are unicellular. For those who are multicellular, they are called moths and mushrooms. They are the multicellular. However, if it's a yeast, it is is just unicellular. One cell, unicellular. Multicellular, many cells. Now, these fungi are good in absorbing nutrients. No? They are powerful. They have powerful exoenzymes that can suck your nutrients. That is why they grow even at low pH. And for them to grow, they need high amount of salt and sugar. Most human fungal infections are nosocomial or occur in immunocompromised states. So they are opportunistic. They are finding their way when it is good for them to grow. I think this is a repeat. Why do we have these things here? Because fungi are needed for you to enjoy this food. No? If you like cheese, you like wine, beer, they all utilize this fungi. Fungi are everywhere. They're everywhere. No? Maybe here, right now, in our room, there are some fungi there in a corner. You just don't know. And even in you are fungi that are naturally occurring. Now, how do we classify these fungi? Of course, when we get to know things, we have to know their classification. They are classified in three ways. They can either be a mold, a yeast, or a dimorph. Dimorphic means it can either be a mold or a yeast. If you are a mold, as you can see, there are two kinds, septate and aseptate and septate. From the word itself, there are divisions, lines that you can see here. It's a continuous one. This is the actual microscopic view of a mold. That's how it looks. See, it's elongated. No? And when you grow it in culture, you will have two types of growth. Aerial and vegetative. If it grows on top, it is aerial. If it grows under, it's vegetative. As simple as that. Alright? And then of course, you have your yeast. As you can see, it's like circular to oblong. That is the shape of them. Alright? And they are single shape. Single spherical shape, as you can see there. And they grow reproductively by budding. So that's one. It goes budding. It will become several forms already. Alright? Next is the dimorphic. The other one. As you can see, there are two things that you can see. You can see the thin hyphal, and you can see the spherical or the round-shaped organism or cell. So that means it has two forms. So dimorphic. Alright, I think that is very common sense, no? Now, these molds, usually, they grow at a high temperature of 37. So if you notice, if you put some food in a warm area, and you just let it there, the moment you come back, you will see some green things on it. Usually you can see this in bread. If you have left a bread out of its expiration date, you will see there are green things on it. These are molds. This is the gross structure of a mold. Alright? Now, sometimes, if you keep it in a 25 degrees centigrade, so this is a little bit cooler, just like the temperature in the room right now, this can invite dimorphics to grow. So if it's very warm, it's the molds that are prone to grow. If it's going to the cold area of 25, it's more of your dimorphic. Alright? Another classification is by their reproduction. Some of it can produce asexually, which is through the spore formation, hypal elongation, and they are called anamorphs. So if they asexually reproduce, they are anamorphs. If they are sexually reproducing, they are telemorphs. Okay? So sexually, telemorphs. If it is a combination, you call it holomorph. There is a sexual and there is sexual reproduction. So, easy. Let us now go to phylum classification. Now this time, if they are classified according to certain characteristics that they have, they are divided into 4. I think this is found in Dr. Gigi. This is exactly from her slides. Ascomycota, Basidiomycota, Glomerulomycota, and Microspara. I know you have read what she has put there. What is, or you have read something about it, I hope. Ascomycota. The mycota itself tells you it's a fungi. The asco is the classification of it, the characteristic of the specific group. So ascomycota, what does it mean? What does it mean in ascomycota? As I have said a while ago, this part of the lecture is supposedly recitation type. But I will not ask you anymore. But if you recite, you get the perfect score. If you don't recite, you get the passing score. And of course, if you are not here, you get a 0 score. Alright? So therefore, what have you learned about ascomycota? What have you learned about this ascomycota?
Fungi that produces ascospores.
Yes, from the word asco. That's why it's ascomycota. Because they're the one with that structure known as ascus. They have the ascus. Alright? And this is through sexual reproduction. They use the ascus to produce more of their kind. If it is asexual reproduction, what do they use? Production of what? Conidia. If it is sexual, it is the ascus part of them. If it is asexual, it is conidia. And in medicine, there are 4 medically important classes of this particular group. What is your name, my dear? Yes, you. Who? Maldavi. Alright. So again, what are the 4 classes that are of medical importance? Meaning, these are the members that can cause diseases. So what are the medically important classes of it? Oh yeah, I know you know this already because I saw this in your Moodle. Anyone from the class? No one? No one have read this? Alright, no one will get a perfect score here, only Magdabi. So these are the 4 classes of medically important ascomycota. We have pneumocystodomycetes, Saccharomycetes, Europhiomycetes, and Soradioriomycetes. The mycetes there is the classes of the fungi. Alright? So how do they differ from each other? Anyone here who knows how they are differentiated? What's the difference between them? The pneumo, the sordario, the urosio, how are they different from each other? Why is the names as such? No one? Oh yeah, because you have a test, no? In virology. So no one bothered to read about this. The first one is named as such, pneumocystis, because it is a protozoan before, Pneumocystis carini. I don't know if you have heard about that. Pneumocystis carini is before a protozoan. However, when they try to study about the organism, they have found out that its cell wall has chitin. So if it has chitin, it must be a fungi. And that is why it was reclassified. So it is now Pneumocystidomycetes, its own class. No, it has its own class. The next one is Saccaro. So Saccaro, the next one is Saccaro. What does it mean? What does it particularly represent? Huh? Do you know? This includes the popular species. What popular species of fungi do we always encounter? Yes?
Yes.
Yeah, what particular species? Yeah, sometimes the doctor will tell you have a yeast infection. What does that mean? What yeast do you have? Yes, Candida. This is the polymorph of Candida. So therefore, if you heard that the patient is having Saccharomycetes infection, and I ask you what infection is the patient having, the patient is having yeast infection. And what is the causative agent? The Candida species. Alright, the first one is Pneumocystis carini. The second one is Candida. How about the third one? The Urasio. What particular organism are under this? The Urasio has a T because trichophyton. The trichophytons are the ones that are under this particular class. The trichophytons which are causing your dermatophytosis. The tinnias that you know. I hope you know them now, huh? The tinea. After this lesson, you should know the tinnias. Then we have the soradario. The soradario. What are the soradario? These are the ones causing keratitis. No? Specifically, fusarium. No? Fusarium. So, I have given you the example, the representative infection of each group. So, in the exam, if I tell you a patient was diagnosed having a Saccharomycetes infection, and I ask you what exactly is the patient having, you should know which exactly is the patient having. Is it a Pneumocystis carini infection, a Fusarium infection, a trichophyton infection, or the Candida infection? So you should know that. That is how I'm going to ask you in the exam in terms of this. This is just under one? Just under ascomycota. We are not yet moving anywhere. So this phylum, Ascomycota, has these specific examples for you. It's a table gotten from your book and also found in your PowerPoint, no? Displayed in your LMS. So here you see the example that are found on these particular groupings. And I expect you to know them. Now, let us now go to the next one, the Basidiomycota. The Basidiomycota is the most common human pathogens. It includes some of your Trichosporon and your Malassezia. Malassezia purpur, it is under this Basidiomycota. Alright, now this one are yeasts. There are most commonly yeasts, which are prominent human pathogens. Basidiomycetos yeast. Then we have Glomerulomycota. Glomerulomycota. They are asexual spores. The Glomerulomycota are asexual spores. Alright? And lastly is the Microspora, which is the smallest organism in this phylum classification. But we are more in this classification. So I just presented to you the different classification of the fungi, but this is our emphasis. The classification of mycosis based on where do they infect. Are they superficial, cutaneous, subcutaneous, systemic, or opportunistic? These are the 5 classifications of mycosis. And your lectures is also according to these classifications. And today, only the superficial and cutaneous will be discussed. Alright? All of this are infecting living cell. A cell that is undergoing the normal processes. These two are infecting dead cell. Only dead cell. So if it's only dead cell that it is infecting, the superficial, we are talking about superficial. What layer of the epidermis will it infect? Yes, only the stratum corneum. It cannot go anywhere deeper. Only the stratum corneum is affected by the superficial infection. And all the superficial infections are here in front of you. We have Malassezia, Tinea, and the Pedras. And they are caused by different organisms. It's only one. You have to remember that. Like Malassezia purpur for the different Malassezia infection. Like Pityriasis versicolor or Tinea versicolor. And seborrheic dermatitis. We have Tinea infections, which is by Hortiae wernicki. Alright? This is only Tinea nigra. Not the other Tineas that you know. Only Tinea nigra is caused by superficial. Then you have white pedra, which is by Trichosporon bigeli, and black pedra by Pedriae hortae. The other Tinea are cutaneous infection. Tinea barbae, Tinea cruris, Tinea, whatever Tinea that is, it is cutaneous. What is the difference? Why Tinea nigra is under superficial and the rest of the Tinea are cutaneous. What is the difference?
Maybe up to stratum basal.
Because?
Maybe it's infecting up to stratum basal.
Yes, it infects the living cell. The cells that are actually having processes still, it can infect up to the dermis. Your 2 layers of the dermis can be infected in the cutaneous infections. That is the difference. Superficial, only the non-living cell. And you all know it is the keratinized layer, the non-keratinized layer, which is known as the stratum corneum. Then the cutaneous is for your other tinea infection. Only tinea nigra is a superficial infection. Let us give you examples. These are the different fungal infection. By the end of mycology, you should be able to identify them. We have here subcutaneous infection and systemic infection, which is basically more serious than your cutaneous and superficial infection. The superficial only affects the stratum corneum. The cutaneous up to the dermis. I hope I have emphasized the difference of the two. And these are the different classification with the example species under each. This is also found in your book, micropara book. Alright? So the superficial, these are the examples of it. The subcutaneous, the opportunistic, and the systemic. Mode of transmission. It has different mode of transmission. It can be through blood, through contact, through food, or through inhalation. However, fungal infections are mostly contact transmission. Skin to skin, touch to touch. Alright? So that is the most common way of infection. The thing about these fungi, they are mostly opportunistic. They only cause diseases in people whose immune system has been weakened. If you have a good immune system, this doesn't have a chance. They will not have a chance, even if you have them already. I know that these 3 pathogenic factors, you have already known them. There is adherence, invasion, and injury. Adherence, meaning they should be able to attach to your cell. Without attachment to your cell, there is no infection. If there is no invasion of your layers, there is no infection. The superficial only invades up to the stratum corneum. The cutaneous infection invades up to stratum to the dermis. And there should be injury. There should be disruption of your normal processes. So that is what makes it pathogenic. Now, how will you be, as I have mentioned, if you are healthy, it will not do any harm to you. Even if there is already your trichopython, no, you touch something with trichopython, if your immune system is good, you will just shy it away. No? So what are the things? These are the several things that can make you prone to it. And I know you have this in your PowerPoint as well. No? One is prolonged antibiotic therapy. Why? Why is prolonged antibiotic therapy a condition that makes you prone to fungal infection? Because when you have prolonged antibiotic therapy, your gastrointestinal system has a natural protective bacteria. If you take too much of your antibiotic, for every single condition you take antibiotic. You sneeze, you take antibiotic. You have a rash, you take antibiotic. If you do that, your natural bacteria are depleted. Depleting them gives no protection to your gastrointestinal tract. So if you eat this you definitely get sick. Because you don't have the good bacteria anymore. So that is why prolonged antibiotic therapy is a no-go. If you are immunocompromised, you have other diseases, you have diabetes, you have cancer, these are states wherein your immune system is down. Therefore, you are really prone to them. Age. What age do we mean here? Extreme age. Those that doesn't have much good immune cells and those that doesn't have yet the immune cells. So very young and very old. Surgical procedure, post-operative. Every time you have a medical procedure, surgical procedure, your immune system are also trying to catch up. Therefore, causing you to be prone to infections as well. Immunosuppressive, from the word itself, it suppresses your immune system. Irradiation therapy, all the same, it's killing your cells. Therefore, giving your immune system a little bit weak. These are all conditions decreasing your immune system. So basically, it is all about your immune system. Alright? These are conditions all referring to your immune system. All the conditions we're going to talk today has microscopic examination as the really diagnostic. Once you see them, that's it. No? You don't need to do culture, serological test, skin test, PCR. It's a waste of money. Mycology is a diagnosis of presentation. What is presented to you, you can diagnose. You don't need any test to confirm. If you need to confirm it, that means you don't trust your clinical knowledge. So let us now start with the superficial mycosis. What are the superficial mycosis? These are the examples that I know is also found in your PowerPoints. The superficials are the malassecias, the nigra, and the pidra, as I have shown to you a while ago. The cutaneous are all the tinea, which are called dermatophytosis. Alright, and of course, on your mycology too, these are the ones you're going to talk about. So let us now start with the superficial mycosis. We have here now pictures of it. In my exam, there's always a picture. Because you have to diagnose them as you see them. So therefore, you better know how they are presented. This is Malassezia purpura presentation. There are blotches, discoloration on the skin. The horte, wernicke, is an encapsulated discoloration. The white pidra looks like dandruff, but they are not. There are nodules attached to each strand of the hair that are white. The black pidra is color black. Thus, black pidra. So Malassezia infections, which is caused by Malassezia purpura. F-U-R. Alright? It is a basidiomycetes yeast, as we have studied a while ago. Basidiomycota. Alright? It's a dimorphic fungi. Alright? And this Malassezia is the one causing pityriasis versicolor. Versicolor. You see the color is different. Varied colors on the skin. This is how you will know that the patient has Malassezia purpura or pityriasis versicolor. Pityriasis folliculitis. As you can see, it's mapped in a hair area. It's in the follicles. That's what it has affected. Then we have, so these are follicular pustules and papules. So it's a map. Let us, this is pityriasis versicolor. You see this area, in most people, they don't have much hair. No? And this pityriasis folliculitis is in the hair area. We have to refer to most people. Because some people here will have hair on their chest. That is not where pityriasis folliculitis will occur. Alright? Only in the hairy area of your body. Then we have seborrheic dermatitis. As you can see, these are white patches. Now, this is still Malassezia furfur. And they are like maps that even if you scratch it out, won't come off. It won't come off. Alright? Usually this one has a genetic predisposition. And sometimes it's about fatty chain acids that are causing this to accumulate on the surface. So these are them in a tabular form. This is your pityriasis folliculitis, your seborrheic dermatitis, your versicolor, your tinea nigra, your white hydra, and your black hydra. I hope you can distinguish them. Alright, so let us now focus on pityriasis versicolor. Where is the versicolor of this patient? In what location is it found?
Eyebrow.
Where?
Supraorbital.
Yes, in the superior, supraorbital area. That is where the pityriasis versicolor, also known as tinea versicolor. This is non-contagious. Most people thought you can give it away. No, you can't. You can't. It is non-contagious. Most of our superficial infections are non-contagious. It's only you that will get infected by it. So this one interferes with your normal pigmentation. That is why you have an either hyperpigmented or hypopigmented. And mostly it affects the neck, the trunk, of course here, it is the face. Maybe that's the one that gets exposed most of the time. The causative agent is of course Malassezia furfur, common in warm and humid conditions or tropical countries. It is frequent in adolescent period. They say that it's because of the high lipid content during this period. When did you have pimples? Adolescent, remember? Because your sebaceous glands are so active. You are so lipid rich. And that's what they love. Remember, fungi loves lipid. That's why this is the age group. Adolescent and young adult. Lipid rich environment. That's what they need. So, Malassezia furfur, it's common. It's common. They are found in healthy skin, especially in your oily area. However, it converts into its filamentous form if the immunosuppression happens. Once your immune system goes down, you can develop it if you have it already in your skin. So, what can lead to pathogenic conversions? One, if you have genetic predisposition. Or you have a relative that has it, then you are prone to have it. If you are commonly in a heat and humid environment, if you are immunodeficient, when you become pregnant, when your skin becomes too oily, because you apply too much oily lotions that can predispose to it. How does pityriasis versicolor looks like? I don't know if you still remember your histology, but is this found in the stratum corneum?
No.
No. So these are the fungi. These are the fungi. No? Those are the fungus. And as you can see, they are in the stratum corneum only. You don't see them you don't have them in the germinale, you don't have in the basale. So therefore, it is really just a superficial infection. So these are all Malassezia purpura, no, in different stages of its development. You better know how do they look like. Because in one exam, I will ask just that this was found in your patient. Which of the following will be possible for your patient to have? So you better know how they look like. You better know how does these organisms look like. So we have here, all of these are Malassezia purpura under the microscope. Alright? So this is just a table showing you the different infection caused by Malassezia, giving you the possible pathogenesis, why it caused the presentation, what you can do to diagnose it, and differential diagnosis for it. But of course, what have I told you about fungal infection? Do we need to confirm it? Do we need to confirm it? No. Please don't ask your patient to spend money for nothing. They will be treated. And mycology is treatment by diagnosis by clinical manifestation. What you see is what you will treat. Alright? So these are the treatments. The good thing about fungal infection, all this azole will work. Whatever fungi it is, it will work. No, the azoles are the maintenance or the main medication for this. And these are over-the-counter preparations that you can advise to your patient. As you can see there is selenium sulfide shampoo. No, you see that the selenium sulfide shampoos in your groceries. So that's simple. You can ask your patient to use it, and eventually, if they use it for 6 months, it will go away. However, if they use it only for a few weeks, it will remain. No? Steroids are not really that advisable unless it is resistant. No? Because you have to lower the immune system. And remember, it all started with a rolling of the immune system. And most of our antifungals are mixed with steroids, with corticosteroids. So as you can see, this is a normal skin, and this is a skin with seborrheic dermatitis. The changes in the sebaceous secretion makes it prone to the infection of seborrheic dermatitis, causing overinvasion of your fungal. So that's why you create your patches, the white plates in your, you still have 20 minutes, on your skin. This is just comparing it to what happens in atopic dermatitis versus seborrheic dermatitis, because patients with seborrheic dermatitis are always misdiagnosed as atopic dermatitis. And in atopic dermatitis, what you only give is steroid. Steroid lowering the immune system. If there's a fungal infection, do you think it will heal? Of course, it will get worse, because you're still lowering down the immune system. Alright, so these are the different treatment. I already gave it to you. It's the same thing. If you have noticed, there's the ketoconazole, miconazole, these are the common preparations you will see. Alright? And it has different. One thing that seborrheic dermatitis sometimes is misdiagnosed for, or the dandruff is diagnosed as seborrheic dermatitis. What is the difference? What is the difference in presentation? Seborrheic dermatitis and dandruff. In dandruff, the plaques are flaky. No, it's flaky. When you hold it, it will crumble. Here, it is patches. It's a patch. It will not be removed even if you scratch it. If you see the hair with a lot of white hair on top, that is dandruff. If you see the white attached to the skin, that is seborrheic dermatitis. Difference, no? And here, it's really oily. No, this one, you will not see any erythema. Here, there will be some erythema because it's an inflammation. Alright, so it's an inflammatory reaction. There is inflammatory infiltration, while in dandruff, there is none. So these are two things that are always misdiagnosed. Atopic dermatitis, dandruff, and seborrheic dermatitis. You should know the difference between the three so that you will not add up to the census of misdiagnosis. In any of our fungal infection, only skin scrapings are the sample that you need. Skin scrapings that will be under the microscope. Sometimes you can do a KOH. No, a KOH, which will show you your thick-walled, round yeast-like cells. This is what you will see in a KOH. As you can see, they are thick-walled, round-bodied yeast-like cells. So that is diagnostic of Malassezia purpura. If you want to culture, no, you have a rich patient, you ask for a culture, Sabarode's dextrose agar or Dixon's agar. The growth will look like this. No? But why do you have to do that? You don't need to do that. This is just discussed to you because you need to know that they exist. But you don't need to have your patient undergo this. Serology, there's none as of now. It is basically microscopic identification, if you want. Alright, management, we have already given you the management for this. The ketoconazole are very effective. These are the common medications that you can advise your patient, whatever fungal infection it is. The topical selenium sulfide, the oral ketoconazole, or the oral itraconazole. Those are the treatment. Let us now go to tinea nigra. This is a tinea nigra caused by Horte Huaniti. They are brown to black macules. They are usual on the palmar aspects of your hand or even on the plantar surface. They are non-inflammatory, they are non-scaly. Alright, this is how their culture will look like. The tinea nigra. It is usually laboratory diagnosis. Again, 10% KOH. This is how tinea nigra looks like. It's still the same. Malassezia purpura. No? And this is the culture still with Sabarode's dextrose agar. That is a misslide. That's not supposed to be there. Oops, and then we missed our slide. We go now to white hydra. Alright, white hydra. This is rare. Seriously, I haven't seen a white hydra. No, I haven't seen it. And it's a yeast-like fungus called trichosporon. That is the causative agent, trichosporon. It is frequent in tropical and temperate zones, though it's not common here. It's found usually in your facial, axillary, or genital hair. So how do they look like? This is the hair, and that is the white hydra. It's like they have grains on their hair. No? So that's how they look like. If you isolate it, this is how they will look like. It is put in a test tube. Under the microscope, that's how they will look like. Alright, so that's white hydra. It is a yellowish nodule, loosely attached to the hair. If you get it out of the hair will break. So don't do that. Your patient will end up with different length of hair if you will cut it off. The effective medicine for this is itraconazole 100 milligram OD until everything is gone. Usually 3 months follow-up is asked of our patient to know if it is effective and the population of the hydra is decreasing. Alright? However, remissions are common. You thought you don't have it anymore, but it will just come back. Now, another is dandruff and white hydra. Oh, you will be diagnosed, okay, use you use the shampoo, anti-dandruff shampoo. And then the patient has been using the anti-dandruff shampoo for the longest time. Still, the hydra won't go away. No? Because it needs azoles. You need your antifungals here. Here, it is just, you just have to do the shampoo. A gentle shampoo to reduce the oil build up. Because that's what happens here. No? If you have a flaky hair, you have a lot of flakes due to the too much oil that was produced by your, remember when you eat, for example, you have a soup that was from maybe beef. Remember, after some time, there will be white things there? That's exactly it is. The lipids getting off of your hair. Alright, so just get it out by doing regular shampoo that will reduce the oil build-up. The black hydra, this time the nodules are black. And this is Hydriae hortae. It's a pigmented hyphae. This is common in Africa, South America, and Asia. But it's not very common in the Philippines. The manifestation, as I have said, is that a dark brown to black nodules on the hair. And this is an example of an actual patient with the condition. This is the microscopic view of your black hydra. And it is cured by terbinafine 250 milligram daily for 6 weeks. The cutaneous mycosis, I will not discuss this to you. These are all the tinea. They're all the same. The name change based on the location. Just the location. So instead of wasting your time now, you still have enough time to read for your exam. The slides here are the same as the slides that you find in Dr. Gigi's presentation. So therefore, you just look at it and make sure that you know about it. This is the only picture that is not found there in your PowerPoint. So get a picture of this. This is the only one. This is Tinea capitis and Tinea barbae. The word tells you where it is. Capitis, no? Of the scalp. Barbae of the mustache area. The rest are the same. This as well is not found on your lecture. The trichophytid reaction. It is basically a hypersensitivity, an inflammatory reaction, a rash. A rash. It is a rash. So you can see there is a rash on the hand, showing you that there are inflammatory reactions happening on the skin. Alright? So that is the whole of the superficial and cutaneous mycosis, as well as introduction of mycology. I wish you luck on your exam. You have still 15 minutes to review for your class. You good luck.